Good morning, fellow Ewing researchers. We're up bright and early this morning. The objectives, this is a remake of the botched Zoom meeting that we held in St. Louis, the bonus session. The recording, the audio and video was really terrible. So I'm going to try to replicate. Unfortunately, we won't have the questions that came from the audience, but I think I have incorporated the answers to all the questions in a revision to this presentation. All right, so the objectives when we're finished, let's know the four types of DNA. That's not a biology hard hurdle at all. We want to know the types of uh, best two types of test results that we can use for as a genealogical tool, and recognize the problem with unphased DNA. So we should have a reasonable appreciation of what unphased DNA means and how we can deal with it. We also want to understand how the triangulation of DNA works, understand how to paint utilizing what I call the eight contributor concept, recognize that the more upstream and or lateral DNA, the better, meaning get as many people tested as you can. Get your dad, your mom, your grandparents if they're living. If the grandparents are not living, get as many aunts and uncles or great aunts and great uncles. Uh, and if none of those are living, then get your cousins tested. The more test uh, results, the easier it is to use DNA as a research tool. And finally, we should understand what centimorgan, that's what the little C stands for, centa, and M stands for Morgan, that it's a measure of how large a match is. And the larger the match, the closer the common ancestor. We'll talk about that in a lot more detail in later charts. All right, so we'll cover Y-DNA, AT-DNA, which is autosomal, X-DNA, and mtDNA. So those are the four. We should understand them a little more thoroughly later on. We'll cover the eight contributor concept. We'll look at the labs and the pros and cons of each of the labs, and I'll give you my recommendation of which one to use. And then we'll look at a few tools, primarily GEDmatch and Painter. We'll also quickly look at WikiTree. And what we want to really come away with is these two things here. Those are the two. How to triangulate, what triangulation means, and how to paint your DNA. The rest of this just gives us an understanding of what it is we're doing when we do these two things. This is what we want to be able to do at the end of this Zoom meeting. So we'll cover GEDmatch, which is where we triangulate, and DNA Painter, which is where we paint. We'll talk a little about WikiTree, and I'll make a recommendation that you use it in injunction with whatever else, whatever other tree tool that you may use. We have 23 chromosomes, one 23 pairs of chromosomes. One of the pair we receive from our mother, and the other one we receive from our father. Chromosomes are named, which are actually numbers, 1 through 22, and the 23rd pair are labeled X and Y. Chromosomes 1 through 22 and the X chromosome, so 45 of the 46 chromosomes that we have recombine during mitosis or meiosis, which is the process that generates the egg and sperm. This mixing and matching is what makes each of us unique. Um, we, we all have two ears, two eyes, two noses, two legs. We don't have two noses, two nostrils. Um, some with hair, some with not. So this is the chromosomes that make us unique. The X and Y chromosomes are sex chromosomes. Females have two X's and males have a Y and one X. The Y they got, the Y is passed from father to son, father to son, father to son, generation after generation, and doesn't mix and match like the other 20 or the other 45 chromosomes or the other 22 and a half pair. It is the only chromosome that does not mix and match. That's what you really need to remember. And therefore, it can be used to identify a family line, male family line, for several generations, actually all the way back to Adam. And we'll deal with that in another video. The average length of our chromosomes are about 2 inches long. And notice that number 1 is the longest chromosome and 22 for the autosomal is the shortest. By far, the puniest is the Y. It is indeed the shortest uh, chromosome. All right. So 1 is longest and 22 is the shortest. Uh, the complete genome, which is all of your DNA, called genome, is about 3 million base pairs long. We'll talk a little more about base pairs in a couple more slides, but think of it as the rungs on the ladder. DNA is a ladder. And so there's 3 billion steps or rungs on that ladder. If you stacked all of the chromosomes within one cell on top of each other, they'd be about 3 meters or 2 meters tall, about 6 foot. So real small. All right, now let's dive into a little biology. We got a little biology and 3 or 4 charts, and then we'll get off the biology, I promise. Um, our DNA is found in every cell of our body, and this is an explosion of a typical cell in our body. It could be a hair cell, a saliva cell, a skin cell, fingernail cell. Um, the only real cells that doesn't have this structure are red blood cells and a couple of other unique. But for the most part, we can say every cell in our body has this structure. It has a nucleus. Okay, it has a nucleus. Um, and inside that nucleus are the X DNA, the Y DNA, and the autosomal DNA. So it looks like a bowl of spaghetti if you looked at it under a microscope. All of these strands of chromosomes are all twisted. And in addition, within each of our cells is mitochondria. Chondria. I have trouble pronouncing that name for some reason. Mitochondria. We abbreviate it as mtDNA, and I'll be using MT because I can pronounce MT. Um, think of this as the battery of this cell. Um, it is what gives the energy to every one of our cells. We get mitochondria from our mothers. So it's passed from mother to children. I have my mom's mitochondria DNA. My children have their mothers. They don't get it from me. You get it from your mothers. Males do not pass it on, females pass it on. And it is much like the Y chromosome. This is not a chromosome, but it is DNA. It is like the Y chromosome in that it's passed from mother to daughter, mother to daughter, mother to daughter, generation after generation, and allows for uh, a way to identify um, earlier ancestors. We'll talk a little later on. It's not a very good tool to use for genealogy, but it is a very interesting. The results can be very interesting to determine your maternal ancestry. Um, I think that's all I want to say about that. Oh no, I don't. So you'll see many cartoons where these chromosomes looks like X's. You see this? That is not signifying the X chromosome. That is representing what the pair, let's use this one here. We'll say it's number one, chromosome number one. That's what it looks like when it begins to replicate, when it begins to duplicate. So when the cells are dividing and making more and more, like skin cells replenish themselves pretty often. Old ones die off and new ones are generated. And during that process, the the mothers and the fathers join together and then they peel apart, and we'll have a picture of that earlier. But anyway, the point is that this is where chromosomes are starting to replicate. That is not designating them as the X chromosome. Now, in this cartoon, the X is signifying the X chromosome. And since it recombines, meaning they're put together and form a replicated copy of it, it is the X chromosome in the process of being replicated. Next to it is the Y. The Y does not look like a Y. It looks just like a little puny thing. And all the other ones here now, you can see they're not using the cartoon as an X. So the number one there is not being replicated. All right. So here we'll call this chromosome one. My wife is in the background laughing at me. Chromosome one. It's getting ready to replicate. Actually, it is replicating, and that's what this shows. How it replicates, it actually separates. All right, but before we get there, let's talk about this um, chromosome. Uh, they are a double helix structure, meaning they're coiled. Uh, and if you untwist it, they resemble a long ladder. And so that's what we'll start thinking about. The chromosomes is an untwisted ladder with rungs. There are four color used in a rung. Rung, rung of our ladder. Those rungs are made up of four acids. I won't try to pronounce them. And by the way, in all the results, they're always represented by their first letter. So A and T for thymine, G guanine, and C for I won't pronounce it. Anyway, A's only bond to T's, and T's only bond to A's. C's only bond to G's, and G's only bond to C's. Okay? So now let's take this twisted double helix, untwist it, zip it apart, and create the left-hand side of the ladder. Let's take the ladder and lay it on its side, and that's how we will start representing DNA. We'll cut a segment of the DNA. This is the entire chromosome 1. We're taking the very last segment, slicing it off. And so that segment has a start and has a stop point, and we'll talk about that a little more at the end. And so from now on, we will only deal with A, T, C, Gs. We know that since A only bonds with T, we don't need to worry about showing this data. We only have to, we know it's an A, a T, a G, an A, a C, C. And so this segment, this is called a segment of chromosome 1 in this case, and we will only discuss the left-hand ladder, the left-hand half of the rungs of the ladder. Just simply reduce the number of A's, a bunch of letters, okay? Um, so this is a DNA segment. It has a pattern of A's, T, G's, A's, C's, G, G, A, T, C, 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 A, G, G. And so if someone else had this exact match, then you would have a match at that location, at that segment of that chromosome. And we'll address that a little more later on. So the Y DNA, we, the father gives it to the son, gives it to the son, father gives it to the son, father to son, father to son, father to son. It does not recombine, meaning it doesn't take a Y from dad and a Y from mom, because mom doesn't have a Y, and mix it together and to create a new unique. It doesn't do that. It stands on its own. And it does mutate, meaning every once in a while there will be a SNP. You don't have to remember this. SNP is its uh, initials. It stands for single nucleotide polymorphism. You don't need to know that. Just know that it's a mutation. And it happens about once every couple, 3 generations. The same is true for the mitochondria DNA. Remember the battery within each of our cells. So you can say that it's because of mom, all work gets done. And we all already know that. If it wasn't for mom's mitochondria DNA in each of our cells giving us energy, and how that mitochondria does that, it takes the sugars um, or ketones, mixes it with oxygen, and it's like a little carburetor. It generates energy for our cells. Um, and when they wear out, we die. Um, so we've talked about 2 of the 4 types of DNA, talked about the Y and the mitochondria. The Y is an excellent tool for use of male lineage, and that'll be covered in a different podcast. Mitochondria, unfortunately, isn't, for a whole host of reasons. One is there's not a lot of it, and there's the mutation. We'll talk. We'll show. We got a graphic. We'll talk about that in a minute. All right. So now we're on the AT version type. This is the 3rd type of DNA. This is a very useful, can be a very useful tool for genealogy. We get all of our ancestors' DNA. So we're made up of a little red and a little orange, a little yellow, a little light blue, purple, green, I don't know what color, and a different color green. And so our makeup now is a bunch of colors. It's what makes us unique. We may be very similar to this person because we get more of, in this case, his DNA. And in this case, your mom got a lot of this person's DNA. And that happens. It's random. And our sisters didn't get the same combination, obviously. Our siblings did not get the same combination, nor did they get the same proportion from each ancestor. Here is the 8 color contributory. Anyway, this, I want you to create a chart, and we'll talk about how to create a chart a little later, that literally does identify this is great grandparents. Got 8 of them, 4 from your dad's side and 4 from your mom's side. I want you to create a color associated with each one of them. Okay, and here is mine. So this is me. I'm sorry, my dad. I'm here. This is my dad. This is his mom, or his dad and his mom and their parents. So my 4 great grandparents: red for Joseph Ewing, pink for Mary Watson, yellow for William Julian, and green for Mary Esslinger. Likewise, on my mom's side, here's my mom. Her grandparents, my great grandparents, are Joseph Philbeck, Cynthia, Jesse, and Rachel. And so I've got a color assigned. This is key. We won't talk about why yet, but just get in the habit of start thinking about, you get DNA from this level, not this level. Obviously we do. We get it from our mom and our dad, but they really got it from these folks. And when we get real um, good at using AT DNA as a research tool, we might start thinking about 16 grandparents, but that, I will tell you, will be down the road a long ways, if ever. 8 is hard enough and good enough. All right, so now we're talking about the 4th type of DNA, X. It isn't as useful as a tool either. So both the X and MT are not real good for a uh, tool uh, to use as a research tool for genealogy. And the reason why, and there's one exception to that, and we'll talk about it when we get down here. The reason is, so this is you or your mother. And so you or your mother get an X from mom and you get an X from dad. Well, the dad got it from his mom only, and she got it from her dad and her mom. And then, so you see that there, this X here has no DNA from those ancestors, or no DNA from those ancestors. Likewise, your mom's doesn't have any from those ancestors, none from that, and none from that, none from him, none from them, and none from them. And so it's so convoluted that it's extremely difficult um, to use it as a meaningful research tool, unless you're a guy. And since on the 23rd pair, we only have 2 chromosomes, an X and a Y, if our matches us on our X, we're pretty certain then we can look in our mother's tree to find out who the common ancestor is. Okay, but beyond that, you can't really use it to determine, is it this leg, that leg, or that leg. You just know that it's somewhere in your mom's side that you're probably related. And the reason I say that you are indeed related, but you may also be related on the dad's side. Always there is a possibility of more than one line going to a common ancestor. Need to keep that in mind forevermore. We'll deal with it a little later on when we start using the tools. And so here's our MT, the final type. So now we've had the Y, the X, the AT, which is a good tool, and the one that we're going to spend most of our time on today. Okay. It is an excellent tool. It's confusing, but we're going to take the confusion out of it. So this one's not very useful. It is indeed anthropologically interesting, and we'll see that in the next chart. It's not very big. There's only 16,000 base pairs. All right, base pair, ladder rungs, same thing. All right, base pairs meaning two base bases that are connected, bonded to each other, which forms that rung. All right, base pair. Use it. I'll use them interchangeably. Rung, base pair, nucleotide. All three of those terms are meaning exactly the same thing. I tend to use nucleotide more often than not, but base pair also. I rarely use rung, but all three of those terms are the same. uh, this DNA, which is indeed in a circle, okay, unlike the chromosomes, which are um, what lines are linear, non-circular. The mitochondrial is literally in a circle, and many times the results are given in a circle. So this is a mutation. These are all mutations. You probably won't have them all, only certain ones of them. And recall that it does not recombine or mix and match. It doesn't. There's only one set of mitochondria, so that it can't mix and match. It is by itself. You get it from your mom, and that's it. It is a specialty test. You have to order it special, unlike the X, which comes with your AT results. Okay. So when you order from 23andMe or Ancestry or one of the main labs, you're going to get your autosomal and your X reported to you. And many people consider that the X is autosomal because it's always with the autosomal results, but it really isn't. All right. So MT is not a very useful genealogical tool, but it is interesting from an anthropological. So here's Eve, and she had descendants that created mutations, and some of those came over. And I believe the B is American Indian. Don't quote me on that. One of those are considered the American Indian, and it may even be the X2. I don't recall. But anyway, if you had the haplo, and these are called haplogroups, just a name of um, classification. If you had this classification, we call that classification a haplogroup. If you had the B, then you're you know that you have indigenous North American ancestry. My mom followed this route, little convoluted. Oops, I missed it here. So she came up this way, and she is an I. She's from the haplogroup I. This haplogroup I later goes to the Isles, Ireland. So this is Scandinavia, Norway, Sweden, in that area. Uh, it later migrates over to Ireland and obviously to the United States, in my case. Um, so having your MT done, I had, and it's not all that expensive, and it's interesting to know um, the 50,000 foot level picture. Uh, okay, so we now know the four types of DNA, the two that are useful in uh, genealogy research, the Y, which is only in the males, and the autosomal, which is from all of our ancestors that makes us unique. That's the multicolored uh, cartoons that we saw earlier. All right. AT, autosomal, is all about matching. To use it as a way to break down a brick wall or find um, more of your ancestors. It's all about matching. So the more you can match, the higher probability is that it becomes a useful tool. Unfortunately, some of those matches are not real. I know. Uh, and that's because the results of those 22 pair, chromosomes 1 through 22 pair, which is 44, one from dad, one from mom, 44 different chromosomes are unphased, meaning that when you get your results, your dads and moms are combined. All right. I'm going to take a little time to see if we can't sort this out. So let's start here. Remember our DNA, we've stripped it apart, and we're not even going to talk about the bottom. And so our results are reported like that. And so let's say this is chromosome 1 again, or 10. We'll use chromosome 10 this time. Chromosome 10. And this one is from dad, okay? Or mom, from mom. And these don't match, so don't let that hurt you. So here we go. We're using the upper rung of mom's DNA, all right, on chromosome 10. And here is the upper rung of dad's. Notice that mom and dad match except for in position 4. Okay, we also call those locus, and all of them are loci. In this position, 10 and 4, mom and dad don't match. Other than that, they match on here and here. Now, this is for illustration purposes only, because the lab, we get nothing that tells us which one, what our dads look, or what our moms look like and what our dads look like. What we get is we get what both of them together look like. And together they look like an AA, a TT, a TT, a GA, a CC, an AA, a TC. And by the way, you could inverse this and put the A up here if you wanted to. We don't know. We don't know if this is mom or this is mom or that's dad or this is dad. All we know is the combination of the two yields us a T and a G and an A and an A and a T and a T and a G and a G. We don't know this, that actually is the mom or the dad. This is what's reported, and we have no clue which one of these is mom or which one is dad. That is also true with whoever we match. Okay? Their results is not reported. Here's your dad, here's your dad's, here's your mom's. It's just simply together. At this location, there are two A's. At this location, there are two T's. At this location, there's two T's. At this location, one of them had an A and one of them had a G. We don't know if it's dad that had a G or mom that had the G. We don't know. One of them did. So what the computer then sees is 13, 1, 2, 3, 4, 5, 6, 7 base pairs or nucleotides or ladder rungs match. And they do. There is a G and an A in our person that we're matching with, and there's a G and an A with us. So that's a perfect match for the computer. The problem is that this chromosome doesn't match either one of these chromosomes. And this chromosome doesn't match either one of those chromosomes. So we don't have a match at all, other than these short little segments, and they're so small that we don't use small segments. We'll talk about that a little later on. So now, not only is our head exploded, now it starts shaking all over the place. Well, keep this in mind. We have a process to help eliminate or reduce this to a small number. Just always be mindful, the smaller the match size, the more probable that it may be a false positive. Geez, hold that thought. We'll deal with this a little more later on. I'm going to blow this up so we can see it, so you can see it a little better on. So you notice this chromosome does not match this one, does not match that one. This chromosome does not match that one, does not match that one. We don't have a match. Now, if one of our parents moved, if we could take this one and put it on the other parent, okay, now we do match. Actually, only want to do one. Um, if we could take this G and move it down here, then we would match on the two mothers would match. But right now they don't match. Got it? I hope so. And I trust me, you probably don't. It took me a while before I finally wrapped my head around this. And I had to make this cartoon. There's not one out there that does a good job of explaining this thing called unphased. And oh, by the way, you can order, it's very, very, very expensive, so where you can get results by each uh, parent. Ancestry's algorithm, they try to deduce, and they do a pretty good job of which one is coming from which parent. And we'll talk about that when we talk about ancestry, but it's not absolute. And it's pretty accurate, but not absolute. All right, so keep this in mind. I think our biology is just about to end. I think I got one more chart. And so there's two terms you'll hear often: identical by state, IBS, and identical by descent. Before we talk about those two terms, let's talk about that we match every human being with more than 98% of our DNA. All right. So when we talk about matches, well, we match every human being more than 98% of our total DNA. Remember, our total DNA is 3 billion rungs, base pairs, or nucleotides. All three have the same meaning. So only less than 2% of that 3 billion is mixed and matched to make each of us uniquely different. We all have two arms. We all have eyes. We all have fingernails. Some of us have hair, some of us don't. That's part of that recombining piece. Um, and there's a recessive gene and a dominant gene in that case. Um, but we all have two dominant genes to give us 10 fingers. All right. That's overly simplified. We match a banana with 55% of our DNA. Whoa, what? Yes, 55%. That's from memory. Maybe 45, maybe 37. I don't know, but it's somewhere around there. It's a big chunk. Uh, they're living. It's a living organism. It has cells. We have cells. I can't remember if they have a nucleus or not. I don't know, but anyway, it's a living cell and it has DNA and it propagates and um, anyway, we match a banana at 55%. So, so when we're talking about matching, we're only talking about this less than 2%. Okay. And that less than 2% is what we call identical by descent. We get it from a common ancestor. Well, we get all of our DNA from a common ancestor. So this is a little confusing, but when we say common ancestor, within, I don't know, 15 or so, 20 or so generations, we can start uh, at this many generations, it's easy to distinguish which ancestor we got the DNA. As we go back, it becomes harder and harder to the point that we say it's matching because of state, as opposed to it can be identified to a common ancestor. All right. So we need a method to deal with this IBD and determine how big a chunk it is that we do share. So there was this guy called Dr. Morgan who looked at a bunch of flies and came up with a road path that his assistant finally refined it, and he named it after his teacher, Dr. Morgan, and it's called Morgan's. We don't use the term Morgan's. That's too small a chunk. We use the term centimorgans or 100 Morgans. And a Morgan is nothing more than 1% chance of mixing and matching DNA. So if this chunk, this segment of DNA has 100% chance of matching and it's 18 base pairs long, it will be 18 centimorgan. I'm sorry, it'll be 18 Morgans. Let's make it 1800. 1800, and if there's 100% probability that there's a recombination or mixing and matching of this segment of DNA, then we would say there is 18 centimorgans. Okay? Enough of that. Just know that a centimorgan is a measure of unit of how much we match. Simple as that. Centimorgan is a measure of how much we match. How large is the match? All right. And we will match our parents around 3,500. Okay. That's what that says right there, 3,485. So, and this study, don't quote me on that, 10,000 people uh, provided their results, and the lowest match to their parent was 2376, and the highest was 3720. The mode is somewhere around 3,500. Most of us match our parents around 30, we just say 3,500, 3,480, 3,490, 3,476, and occasionally 3,508. So we match our parents in terms of centimorgans 3,500. We don't know what a kilometer, most of us don't know what a kilometer is, but if somebody said it's about 15 kilometers down the road, we know it's a unit of measure of distance. This is nothing more than that. We match 3,500 centimorgans with our parents. We match about half of that much with a single grandparent, right? Because we have four grandparents. Well, wait a minute, Ewing. Well, we, we, we only deal with one of the chromosomes. So in chromosome one that we got from dad, we match him 3,500. The chromosome one that we got from mom, we match her 3,500. We don't match the two of them 7,000. We simply deal with one line or the other. And so our grandparent, my dad's dad or mom, it's just this one chromosome. They right now will assume that they do not match my mom, which is not that uncommon, especially if your ancestors lived in the United States pre-Revolutionary War. We'll set that aside for a moment. So I match my dad at 35. I match his dad or mom at 17, about half. Okay. And I match my grandparents' parents about half that much again. So every generation, the DNA cuts half and half and half. So therefore, when you go up the tree, it gets so small, and we say seven. When it gets smaller than 7 centimorgans, we ignore it because we're going to get more false positives than our heads will be able to play with. All right, you got that? And so your child also matches because a child parent, right, is 30. I match my dad 3,500. He matches me 3,500. All right. So parents, children, 3,500. Parents, grandparents, half that. Children, great grandchildren, one quarter of that. All right. So it just cuts in half. You got it? Now, what this chart is also good for is if you find someone around 27, then you're looking at a third cousin two-time removed, or let's see, where else? All right, third cousin two times removed. Yep, yep. See any more 30s? But it can range between 0 and 78 because of the ram ram randomness. The randomness of that mixing and matching. Is very possible that our second cousin and me don't share any common ancestor, even though, I'm sorry, any common DNA, even though we do for certain share a common ancestor. The randomness of it, he he or she did not get the colors that I got. He got his own colors and I got my colors, but we don't share any common colors. All right, I think you're following that. The takeaway here, the larger the number, the closer the relative. You can't get much bigger than 3,500. So if you see you're matching someone at 3,500 and their name ain't your dad or mom's name, oops, you're either adopted or you got switched out at the hospital, which has actually happened, um, or there was some sort of NPE, non-parental event. Okay, another code for hanky panky. So don't get your DNA tested if you don't want to find out any answers. This simply chart, and I won't spend much time on it. There is some fluctuation depending on which lab you use. It's not a perfect reading. Remember, this is 39 or 3 billion uh, base pairs, rungs. And so the exact reading ain't there. And to have a little noise, there is a little noise. That's the whole purpose of this meeting. Um, Here are the big labs, the most common, 23andMe. I don't know much about it. I don't use it. Good on you if you do. I do use Family Tree, especially for Y and MT. It's an excellent place to get those done. So Y-DNA and mtDNA. It will also do the AT. Don't use it to get your AT done. Ancestry owns the market on AT. Use Ancestry. You can get it on sale every once in a while for 49 bucks, so you can't beat it. This says it's 69 bucks normal. It goes on sale for 49 thereabout. Use Ancestry. There are some downsides on Ancestry. I'll talk about that in a minute. MyHeritage is trying to take market share away from Ancestry, doing a pretty dang good job of it. But again, remember autosomal, and I'm really just talking about autosomal here. Autosomal is about matching. All right. That's what we're trying, if we want to use it, not to determine whether or not we should be wear lederhosen or kilt. If that's the reason you're having it done, it doesn't matter. Do it wherever. But if you really want to use your DNA as a research tool for your genealogy, then I recommend using Ancestry and then doing something with that. Living DNA, again, I don't know much about it, but it is up and coming. And again, they all get competitive during. You can go to this website, and there's 3 or 4 pages of comparisons. All right, and depending on what you may be trying to achieve, in addition to um, a genealogy tool, one of those labs may make more sense to you than the others. But now what I do, and this is a great place to talk about it, I test here, and for 10, I think it was $10, don't quote me, it's cheap. I take these results and I upload them to my heritage. They allow you to do that. And I take them and upload them to my family tree. And so I get to match over here, I get to match over here, and I get to match over here. I don't get to match here, and I don't get to match here. We'll talk about how we can fix that in a minute. The terrible thing about Ancestry, their algorithm is proprietary, is that they won't tell you what the start and stop. Remember, we talked about that earlier, and what the values are, ATTCG, ATCTG. They don't tell you the segment, nor the values, nor the chromosome that you match. They only tell you how many segments and how big. And later, because we want to learn how to map, you must know where the segment started, where it ended, how long it is, and the values. All right, so we'll talk about that a little more. And we'll do it actually right now. So I recommend testing here and then uploading it to a third party called GEDmatch. It's free. And get there are people who tested here have uploaded. There's people here who have uploaded. There's people here who have tested at 23andMe and uploaded. And there's people who have tested living DNA uploaded. And so now we can start matching with people from the other labs. All right. Remember, it's all about matching. And we want to match to as many people as we possibly can. Now, not everyone from MyHeritage uploaded. So that's the reason you want to download your results from Ancestry to MyHeritage. That's the reason you want to download uh, but you can't. Ancestry won't allow you to upload family tree to them. They won't allow you to upload MyHeritage. They won't allow. So that one, there's two reasons. One is they don't allow uploading, and two, they have by far the largest database to compare against. A, B, so that's A and B. They won't allow you to upload A. That's reason number one. Two, they have the largest matching database. And three, they have the most accurate algorithms for matching. It's not a pure science, right? Remember, we got this phased and unphased, and there are several other nuances that is graduate level, not even advanced. It is not even the uh, if we're a freshman now, it's not even sophomore. We're talking PhD level stuff. We won't even address it. We don't care. Too noisy. Overly simplified. How do they test? They take the DNA that we give them, they cut it in little segments. Think of it they putting it on a old type microscope that's got tubes. They stick it in. First of all, they separate the rung, the hydrogen bonds. They make that ladder. They figure out which the bottom, which they will call the start, and the end, which we'll call the top of the ladder. There's a way they determine that. And then they make multiple copies of it, millions. They attach a means to identify the start and the end. They put some chemicals or uh, dyes, if you will, in there that react to the A, the T, the C, and G, and give each one of them a unique color that later that the computer reads. And so it's reading this color, purple, as an A. This is a yellow. It's reading it as a T. This is a C, and this one it's red, two G's, right? And so now it's got this GGACT, GATCT. That's how the testing is done. Enough of that. We don't need to know it, but at least you have a notion. All right, GEDmatch. We're going to take this video will end here. I will now, so we've, at the end of this, we should know the four types of DNA, which of the two, Y and autosomal, is good uh, for for using them as a research tool, that AT or MT, mitochondrial, and uh, X, those two types, not too good, still interesting, can be used. Um, so we're now, so we've talked about the DNA, we've talked about the labs, we've talked about the importance of matching and what is being matched, those rungs, nucleotides, or base pairs. Remember, all three of those have the same, they all are synonymous: rung, base pair, nucleotide. So we're looking, we want to find people who have matches in a long enough string that eliminates the false positives of unphased DNA. If you got that, not watch it again. You'll get it after second time. So the next video will actually start using the data. We'll start looking at the data and begin to uh, identify the steps that will allow us to triangulate and paint. Remember, that's the two things that we are trying to get to. Know how to triangulate, and know then after we've triangulated, know how to paint it. All right. So that will be the next version. Thank you all for putting up. Leave some comments. So the next time, as negative comments are fine. That way, whatever it is, I can improve upon the next version of this. All right. Bye, all you Ewings. We're out of here, I think. I'm sharing. And